Welcome to another episode of Your Old Anatomy Professor. Today, we're going to review cells. Now, I know you weren't very excited about cells when we studied them in A&P 1, but the first chapter of your textbook reminds you that the cell is, in fact, where all diseases start, right? It's a disruption of cellular structure or function that is, in fact, at the root of all diseases. Right? Think about it. For example, many tissues or cells might be examined during illness. That's what a pap smear is. It's looking for deranged cells. Or a lot of the biochemical tests, blood tests, urine tests, they're looking for changes in the physiology at a cellular level. Remember, this cell is trying to maintain homeostasis, right? That balance of its internal environment, even when the surrounding environment might change. And any disruption to that, a failure of that homeostasis, that's what's going to lead to illness. So let's take a little look at the cell. Remember, the plasma membrane. This is that phospholipid bilayer that encloses the cell. It is studded with proteins. Some of those are glycoproteins, right? This is trying to show a sugar protein mixture. That would be a glycoprotein. The big deal with the plasma membrane, with the cell membrane, you might recall that its function, that's right, selective permeability. This is the barrier between the inside of the cell and the outside, right? Intracellular fluid, extracellular fluid. It's the plasma membrane that dictates when water, ions, nutrients, gases come in and out of the cell. Damage to the plasma membrane is going to be a problem. You might see that show up as either cellular edema, swelling, if water moves into the cell, or potentially something like cellular dehydration. Either one of these is going to be damaging to organelles. Now, one of the most important proteins that you'll find in that plasma membrane is the famous sodium-potassium pump. You may recall that this is a pump, meaning that it's moving against its concentration gradient. It's using active transport, right? So we're using energy in order to push 3 sodium out of the cell and 2 potassium into the cell for each ATP used. One way that I remember this is, if you'll recall, potassium ends up being higher in higher levels in the intracellular fluid. So you can remember that the sodium-potassium pump is bringing it there. Remember, resting membrane potentials, the cell is actually leaky to things like sodium and potassium, and we're using this pump in order to reset that. Okay, let's move on into the cell and take a look at a few of our organelles. Okay, our favorite, the mitochondria, you may recall, is who produces ATP. And it does that through aerobic cellular respiration. Now, what you're going to see is a lot of disease states involve hypoxia, low oxygen levels, in which case we can't use that aerobic process anymore. And in fact, the cell is going to do anaerobic respiration, fermentation, and make lactic acid as a byproduct. That happens outside of the mitochondria in the cytosol. Another thing to remember with the mitochondria is that it has its own DNA. So mutations to that DNA may be involved in things like aging and certain other chronic diseases. How about these lysosomes? We didn't focus on them a whole lot, but they actually become important in a few different disease processes. So remember, this lysosome, it is a membrane-bound organelle filled with digestive enzymes, things like proteases, lipases, lysozyme. And these are meant to either break down like worn out cell parts, maybe other organelles, or to break down things that the cell has brought in through endocytosis, right? That engulfing foreign objects. So for example, macrophages have lots of lysosomes in order to break down foreign bacteria, microbes, that sort of thing. Now, when a cell dies, this lysosome is actually going to spill open and lead to what's called autolysis. It's going to break down the remaining portion of the cell as all these enzymes just spill everywhere. In fact, you'll see this with heart attacks, right? In a myocardial infarction, the dead cardiac cells, those muscle cells, the lysozymes are going to digest them, and we're going to spill enzymes into the bloodstream. That's actually a really important test to see if someone has in fact had a heart attack. There's also some diseases, for example, Tay-Sachs disease is when you actually don't have enough of these enzymes or any of these enzymes in the lysosome, and so substances accumulate. In Tay-Sachs, there are lipids that accumulate, leading to neurodegeneration. Endoplasmic reticulum surrounding the nucleus here. You'll remember there's smooth endoplasmic reticulum, which makes lipids, good. And then the rough endoplasmic reticulum, it's studded in ribosomes. And we remember ribosomes produce proteins, very good. And so the rough ER is also involved then in protein synthesis. You may hear about something called ER stress, and that's going to be problematic in several disease states. Basically, the endoplasmic reticulum being overworked, and this can lead to a variety of cellular problems. Let's see, who else? The Golgi apparatus, that's a fun one. It's taking all those proteins, packaging them, processing, secreting them, right? We see this little packet coming to the surface here. Okay, last organelle, super important, the nucleus, right? The nucleus houses DNA. We'll talk about most of those problems of the nucleus as genetic disorders. So that's in a later chapter. But this is a great time to recall that DNA codes for a protein, but in order to make that protein, we need to go through the process of transcription to make messenger RNA. That messenger RNA can leave the nucleus, go to the ribosome, and undergo translation, where the messenger RNA code is read and a polypeptide chain, which then becomes a protein, is produced. So there you have it. You didn't think those organelles were that exciting, but they're coming back in a lot of different disease processes.